11th Biology. Multiple choice question and answer with explanation. Multiple choice question and answer with explanation. From the following biology topics, list the principles and identify the apparatus used in the techniques of fraction, differential staining, centrifugation, microdissection, tissue culture, chromatography, electrophoresis, and spectrophotometry. Centrifugation. Question 1. What is the fundamental principle behind centrifugation? A. Magnetic separation. B. Density-based separation. C. Electrophoretic mobility. D. Temperature gradient. Answer. B. Density-based separation. Explanation. Centrifugation separates particles based on differences in density. Heavier particles move outward faster during centrifugation, leading to separation. Question 2. Which apparatus is commonly used for high-speed centrifugation in a laboratory setting? A. Microscope. B. Pipette. C. Centrifuge. D. Spectrophotometer. Answer. C. Centrifuge. Explanation. Centrifuges are specialized machines designed for high-speed centrifugation, enabling efficient separation of substances. Chromatography. Question 3. What is the principle of chromatography? A. Magnetic attraction. B. Affinity for specific antibodies. C. Partitioning between mobile and stationary phases. D. Electrophoretic movement. Answer. Partitioning between mobile and stationary phases. Explanation. Chromatography separates compounds based on their differing affinities for a mobile phase. For example, solvent. And a stationary phase. For example, column packing. Question 4. In thin layer chromatography, what is the stationary phase? A. Liquid solvent. B. Paper strip. C. Solid silica gel. D. Gas phase. Answer. C. Solid silica gel. Explanation. In thin layer chromatography, the stationary phase is a solid silica gel or similar material coated on a plate. Electrophoresis. Question 5. What is the main principle behind electrophoresis? A. Separation based on size. B. Separation based on density. C. Separation based on color. D. Separation based on temperature. Answer. A. Separation based on size. Explanation. Electrophoresis separates molecules based on their size and charge, with smaller molecules migrating faster. Question 6. Which technique uses an electric field to separate DNA fragments based on their size? A. Centrifugation. B. Microdissection. C. Tissue culture. D. Gel electrophoresis. Answer? D. Gel electrophoresis. Explanation. Gel electrophoresis separates DNA fragments based on size as they migrate through a gel under the influence of an electric field. Microdissection. Question 7. What is the primary purpose of microdissection in biology? A. To culture cells. B. To isolate specific tissues or cells. C. To measure light absorption. D. To separate DNA fragments. Answer. B. To isolate specific tissues or cells. Explanation. Microdissection is a technique used to precisely isolate specific tissues or cells from a sample for further analysis. Question 8. Which tool is commonly used for microdissection? A. Centrifuge. B. Microscope. C. Spectrophotometer. D. PCR machine. Answer? B. Microscope. Explanation. Microdissection is typically performed under a microscope for precise targeting. Tissue culture. Question 9. What is the primary objective of tissue culture in biology? A. To isolate DNA. B. To grow and maintain cells or tissues outside of their natural environment. C. To separate proteins. D. To perform electrophoresis. Answer. B. To grow and maintain cells or tissues outside of their natural environment. Explanation. Tissue culture is used to cultivate and maintain cells or tissues for research or medical purposes. Question 10. Which apparatus is commonly used for tissue culture in a controlled environment? A. Microscope. B. Incubator. C. Centrifuge. D. Spectrophotometer. Answer. B. Incubator. Explanation. An incubator provides controlled conditions of temperature, humidity, and gas concentration for tissue culture. Spectrophotometry. Question 11. What does spectrophotometry measure in biological samples? A. DNA sequence. B. Absorption of light at specific wavelengths. C. Cell count. D. pH level. Answer? B. Absorption of light at specific wavelengths. Explanation. Spectrophotometry measures how much light is absorbed by a substance at specific wavelengths, often used to determine concentration. Question 12. In spectrophotometry, what does a higher absorbance value indicate? A. Higher concentration of the substance. B. Lower concentration of the substance. C. No concentration changes. D. Higher temperature. Answer. A. Higher concentration of the substance. Explanation. A higher absorbance value indicates that more light is absorbed, typically correlating with a higher concentration of the substance being measured. Fractionation. Question 13. What is the primary purpose of fractionation in biology? A. To culture cells. B. To separate molecules based on size. C. To amplify DNA. D. To perform electrophoresis. Answer? B. To separate molecules based on size. Explanation. Fractionation is a technique used to separate complex mixtures of molecules based on their size. Often used to isolate specific components. Question 14. Which apparatus is commonly used for fractionation? A. PCR machine. B. Spectrophotometer. C. Ultracentrifuge. D. Microscope. Answer? C. Ultracentrifuge. Explanation. Ultracentrifuges are specialized machines designed for high-speed centrifugation and fractionation of particles based on size and density. Differential staining. Question 15. What is the main principle of differential staining? A. Separation based on size. B. Differentiation of different cell types or structures based on staining characteristics. C. Separation based on density. D. Measurement of absorbance. Answer. B. Differentiation of different cell types or structures based on staining characteristics. Explanation. Differential staining uses specific stains to highlight different structures or cell types in a sample. Question 16. Which staining technique is commonly used to differentiate gram-positive and gram-negative bacteria? A. Hematoxylin and eosin staining. B. Giemsa staining. C. Gram staining. D. Immunofluorescence staining. Answer. C. Gram staining. Explanation. Gram staining is a differential staining technique used to distinguish between gram-positive and gram-negative bacteria based on cell wall characteristics. Electrophoresis. Question 17. In gel electrophoresis, which particles move towards the positive electrode? A. Proteins. B. DNA fragments. C. Electrons. D. Water molecules. Answer. B. DNA fragments. Explanation. In gel electrophoresis, negatively charged DNA fragments migrate towards the positive electrode. Question 18. What is the primary purpose of electrophoresis in molecular biology? A. To isolate specific cells. B. To separate DNA, RNA, or proteins based on size and charge. C. To culture tissues. D. To measure light absorption. Answer. B. To separate DNA, RNA, or proteins based on size and charge. Explanation. Electrophoresis is used to separate macromolecules based on their size and charge, which is critical for DNA, RNA, and protein analysis. Question 19. Which of the following is a common technique used during fractionation to separate macromolecules? A. Magnetic resonance imaging. B. Ultracentrifugation. C. Polymerase chain reaction PCR. D. Immunofluorescence microscopy. Answer. B. Ultracentrifugation. Explanation: ultracentrifugation is commonly used during fractionation to separate macromolecules based on size and density. Question 20. What is the primary goal of fractionation in cell biology? A. To perform differential staining. B. To culture cells. C. To isolate and purify specific cellular components. D. To measure the absorbance of chemicals. Answer. C. To isolate and purify specific cellular components. Explanation: fractionation in cell biology is used to separate and purify specific cellular components for further analysis. Question 21. Which type of chromatography relies on the binding of target molecules to a stationary phase containing antibodies or ligands? A. Affinity chromatography. B. Gas chromatography. C. Liquid chromatography. D. Thin layer chromatography. Answer. A. Affinity chromatography. Explanation: affinity chromatography uses specific interactions between target molecules and immobilized ligands or antibodies. Question 22. In high-performance liquid chromatography HPLC, what is the mobile phase? A. Solid, gel. B. Liquid solvent. C. Gas. D. Electrophoresis buffer. Answer. B. Liquid solvent. Explanation: in HPLC, the mobile phase is a liquid solvent that moves through the column to carry analytes. Question 23. What is the primary application of spectrophotometry in biology? A. Separating proteins. B. Measuring light absorption by molecules. C. Growing cells. D. Isolating DNA fragments. Answer. B. Measuring light absorption by molecules. Explanation: spectrophotometry is used to measure how much light is absorbed by molecules at specific wavelengths, which is valuable for concentration determination. Question 24. Which unit is commonly used to express absorbance values in spectrophotometry? A. Volts. B. Degrees Celsius. C. Nanometers. D. Optical density. Odd. Answer. D. Optical density. Odd. Explanation: absorbance values in spectrophotometry are often expressed in optical density. Odd units. Question 25. Which type of differential staining technique is commonly used to visualize the structure of bacterial endospores? A. Gram staining. B. Zeil-Nielsen staining. C. Acid-fast staining. D. Endospore staining. Answer. D. Endospore staining. Explanation: endospore staining is specifically used to visualize the endospores within bacterial cells. Question 26. In differential staining, why do different cell components stain differently? A. Due to temperature variations. B. Due to differences in cell shape. C. Due to variations in cell size. D. Due to differences in chemical composition. Answer. D. Due to differences in chemical composition. Explanation: differential staining relies on differences in the chemical composition of cellular components to achieve contrasting staining results. Question 27. What is the primary purpose of microdissection in genetics research? A. To culture cells. B. To isolate specific DNA regions or genes. C. To measure light absorption. D. To perform gel electrophoresis. Answer. B. To isolate specific DNA regions or genes. Explanation: microdissection is often used to isolate specific DNA regions or genes for further analysis in genetics research. Question 28. Which tool is commonly used for precise microdissection of tissues or cells? A. Pipette. B. Centrifuge. C. Laser microdissection system. D. Microscope. Answer. C. Laser microdissection system. Explanation: laser microdissection systems offer precise targeting and isolation of specific cells or tissues. Tissue culture. Question 29. In tissue culture, what is the purpose of using a controlled environment? A. To culture tissues at varying temperatures. B. To culture tissues without any controls. C. To maintain optimal conditions for cell growth outside of their natural environment. D. To culture tissues in open air. Answer. C. To maintain optimal conditions for cell growth outside of their natural environment. Explanation: tissue culture relies on controlled environments to provide optimal conditions for cell growth. Question 30. Which of the following is a common application of tissue culture? A. Identifying proteins. B. Growing cells for research or medical purposes. C. Separating DNA fragments. D. Measuring light absorption. Answer. B. Growing cells for research or medical purposes. Explanation: tissue culture is used to cultivate and maintain cells or tissues for research and medical applications.